My name is Hamza Fawzi. I'll be the chair for the second intense session of the day. That's a session composed of 6 student talks. Each talk is 5 minutes. And so each student has also a poster corresponding to the talk that they're giving. So we won't have any question after each talk. Instead, you'll have the possibility to ask questions during in the break afterwards. So at the poster of each student. So these are the students of the CCIMI, by the way. So the first talk will be by Lisa Kreusser, and she will talk to us about ODE and PDE modeling of biological networks.
Thank you very much for giving me the opportunity to present some of my recent research here. So today I'm going to talk about one of the projects of my PhD, which is on biological network formation. More specifically, we are interested in modeling different biological networks, and it turns out that in terms of the math behind it, it's actually quite similar whether you consider leaf venation or blood circulation or the transport of neurons in neural networks. In order to describe those biological networks we're going to consider, we're mainly interested in two quantities, namely the pressure and the conductivities, to describe our network, which is set on a graph structure. So we assume that there are a certain number of nodes given which are connected by some vertices, some vertices which are connected by some edges, and we can kind of regard those edges as vessels where our fluids are transported. And the fluid transport is depends on the underlying pressures on each of those vertices, and in particular we're interested in the conductivities. This basically means how much of the fluid is transported along each of the vessels, and we're interested in seeing the underlying structure. So it will turn out that along some of the vessels a lot of the fluid is transported, whereas along other vessels nearly none of the fluid is transported. And in order to see how much is actually transported along the different vessels, we consider the flow rate, which is denoted by Q as the product of the conductivities and the pressures. Since we are interested in the evolution over time, it's quite useful to consider some type of ODE or PDE system in the continuum case. So in order to describe this, since we are interested in describing network formation, we can use some well-known rules from electrical engineering, actually, where Kirchhoff's law has been used for many, many years, and to try to adapt the quantities used in those descriptions and adapt it to our biological network formation problem. So this turns out to be the Kirchhoff law as described in here. And since we also need to find a formulation to describe how the concentrations behave over time, we consider this energy functional given in here. So this consists of a metabolic term as well as an activation term in here. So as you can see, this results in a coupled system. So above here is a linear system. Assuming that the concentrations C are given, we have a linear system for the pressures P, coupled with this energy cost functional, which has to be minimized with respect to C. Unfortunately, this linear system doesn't have a solution all the time, so we have to make sure that a solution actually exists. And one can show that if you consider the graph associated with all conductivities being positive, this is a necessary and sufficient condition for existence of solutions of those pressures P. And once we have those pressures P, we can plug them into our energy functional down here and minimize with respect to the concentration C. And in fact, we were able to show that there exists a unique solution consisting of the concentration C and the pressure drops delta P. Obviously, there is no unique solutions for the pressures P, as they, in all those equations, we always have the pressure drops, so you can always scale by a constant and you will still get a different solution. A second step, which is really interesting in terms of modeling, is that if you consider finer and finer structures, this means we want to reduce the length of the edges and introduce larger numbers of vertices. This means that we are interested in finding an associated continuum formulation. And we could actually prove rigorously that this is the associated continuum model which corresponds to our discrete model. So we have some Poisson type equation here, which is the constraint of a continuous energy functional. And of course, the quantitative behavior of this system is also of great interest, so one can do a lot of numerical simulations in order to see how the different parameters in this model depend on the steady states we obtain in the end. And you can see that, for instance, the parameter gamma in here plays a crucial role, because if gamma is rather small, we get some tree-like stationary states, whereas for gamma larger than 1, one gets a full network structure. And you also can prove this rigorously, that you have this phase transition. And besides, as motivated by the slide before, the question arises, if, for instance, we start with a graph containing a lot of loops, whether those loops vanish over time for the case gamma being small enough. And indeed, this happens. So here you can see different loops as initial conditions. And in fact, the initial conditions are rather similar, but still we get some kind of different associated steady states. So this shows that our energy functional constraint by this linear system in the discrete case has a large number of local minima, so we can't guarantee the uniqueness of minima. So I hope I could give you a short overview about my research. So we got some analytical as well as some numerical results on this biological network formation problem. And right now we are also working with biologists at the Sainsbury lab who will give us some data so that we can specifically for the case of leaf venation improve the model and make some simulations for them. Thank you very much for your attention.